Greetings, everyone. Welcome to our 14th session of the EMN Webinar series of this year. So my name is Pinto, and I'm a postdoc researcher at REQUIMTE LAQV ISEP School of Engineering, Polytechnic of Porto, from Portugal. And I will be hosting this webinar with my colleague Thomas Dussarrat from the Bielefeld University from Germany. And I hope you will be all connected with us and enjoying this webinar. So on behalf of the EMN Committee of the Metabolomic Society, I would like to thank you for joining us, and I hope you all enjoy this webinar. Also, I would like to encourage you to follow EMN and Metabolomic Society online on our LinkedIn and Twitter X pages. So please feel free to follow us and connect with us online. Also, we will make here available the EMN email if you would like to contact us or make any additional question. Please feel free to do it. And without further delay, I am very pleased to introduce you to Professor Dr. Mingxun Wang and Reza Shanheh. I apologize for any pronunciation mistake on my behalf. And they will talk us about illuminating metabolomic dark matter, reshaping how to mine and reuse big mass spectrometry data. And to introduce Professor Dr. Mingxun Wang, is an assistant professor in the Department of the Computer Science at the University of California, Riverside. His lab develops computational methods to organize, visualize, and analyze small molecule mass spectrometry data. He received his B.S. summa cum laude in computer engineering from the University of Illinois Urbana-Champaign in 2009. He earned his PhD in computer science from the University of California, San Diego, under the supervision of Professor Nuno Bandeira in 2017. He completed his postdoctoral training in 2022 under the supervision of Professor Peter Dorstein at the University of California, San Diego. I'm delighted to introduce you, Professor Mingxun Wang, and please, Professor, the floor is yours.
Thank you, Diana. Let me share my screen. It's also, I just want to, it's a small joke. I've actually never heard, so my advisor, Nuno, he's also Portuguese. And I've never heard another Portuguese speaker say his name. So I know I pronounced it reasonably correct when I was a student. So that was fun.
Thank you.
But thank you for the kind invitation and this opportunity to speak. And so I'm excited to present some of the work that we've done over the past couple of years, as well as highlight some of the new tools that we've developed and published on just this past year. And so it's mostly you'll see it in the session after my talk with Reza, the hands-on. The students do all the work, so he's the one to ask the questions to. But we're very excited about it. But this talk is meant to give a little higher perspective about some of the broad approaches and the computational intuition on what makes it all work and possible. And so yeah, happy to get started. And so first, I just want to make sure these are my conflict of interest disclosures. And I think to this audience, we don't really have to go into details of why is metabolomics cool, where can we apply it. This is just kind of a sampling of the areas where I've been fortunate enough to have some great collaborators and apply our tools from metabolomics and the gut microbiome all the way out to the ocean and understanding chemical ecology of different microbiome communities, as well as bringing it back to the clinic and thinking about drug discovery and things like that, and even as fun and unique as forensics. So that was a few years ago, looking at those kinds of data. And so broadly, we view metabolomics and also the computational portions as a tool that has many different different nails that we can hit with that hammer. And so again, I want to just frame this as as the underlying analytical question that we want to look at is we may have some sort of biological or environmental sample, and we want to use a mass spectrometer as one of our analytical tools. And at the end of the day, we want to get out some sort of chemical structures that are what is in the sample and how much is in this particular sample. And so then chemists can start reasoning about why this matters, and biochemists can kind of figuring out how this relates to broader biological question. Now, one of the reasons that we use mass spectrometry in this particular application is high throughput. We can measure tens of thousands, 100,000s of molecules per hour. And so that's the high throughput aspect. It can be quantitative if we do the right experimental setups. And also what's exciting to us is it's very versatile, right? We can look at huge number of different compound classes. You might have to change your setup in different ways, but broadly we can look at peptides, lipids, sugars, nucleic acids, metabolites, or different types of metabolites on a single platform. And this is in difference from nucleic acids that, while powerful to look at sequencing, you're basically looking at with sequencing only a single biomolecule. And so this is not that we actually run any of the mass spectrometry, but we really appreciate the versatility of such a platform. And so at the end of the day, what we're doing with these mass spectrometers is again, they're just very fancy scales. And so, for example, here on the right-hand side, we have a caffeine molecule. We can weigh it with a mass spectrometer. But the problem with us simply weighing it on a mass spectrometer is that we may not be able to resolve different isomers, right? The different organizations of the atoms. At best, we may be able to get a chemical formula and for the composition of the metabolite. But to distinguish between multiple isomers, as we've shown here, just by weighing it, it's very, very difficult. Intact, it's very, very difficult. And so as a technological background, one of the things that we do is that we do tandem mass spectrometry. And so very briefly, very high level, is that we add a little bit of energy to these molecules and they fall apart into some pieces. And so each molecule will fall apart, different molecules will fall apart differently, depending on how the atoms are configured in the 2D structure. And then what we can do again is with the mass spectrometer, we can measure the masses of these pieces. And again, we'll be using the terminology throughout the presentation of tandem mass spectrometry. And so we can see here, a lot of us are familiar with this, but on the x-axis is the mass of the pieces, and the y-axis is the relative intensity of each of the pieces, and this constitutes a tandem mass spectrum. And this will be kind of the basic fingerprint for us to think about when we measure each analyte in the mass spectrometer. And so just to round out some of the terminology, this is more for the kind of computer science people who are not super familiar with this, but we can weigh the particular molecule and we have its precursor mass. So we know what it weighs as a whole. Then when we undergo fragmentation, we end up with a set of mass and intensity pairs, and we have multiple of these that constitute the tandem mass spectrum or the MS2 spectrum or the MSMS spectrum, kind of all synonymous. But we have that's the relationship that we have between the data. And so again, if we take a step back and think of where we started, we have this biological or environmental sample that we carry over to the mass spectrometer and we do tandem mass spec analysis. But instead of giving us a set of structures that we can reason about from a chemical or biochemistry perspective, we end up with a ton of tandem mass spec, hundreds, thousands, tens of thousands, depending on the size of your experiment. It can end up easily in the millions. So it's a lot of data. And so we want to fundamentally ask this kind of question. Once we measure the tandem mass spectra from the sample, we want to change this and convert this over to this chemical space. There's that last bridging step that we're missing here. And so you want to be able to ask that central analytical chemistry question, what are the molecules in my spectrum? But what we have in terms of data is a big set of tandem mass spectra. So just to give us an idea of our ability to annotate these things, back, when we started started my career, at least my graduate career, and we built some of these systems, we could identify in 2014, and when we measured it, less than 1% of all the tandem mass spec in these public experiments. And so it's this just simply less than 1%, but it's kind of hiding a lot of this. It's significantly less than 1%. But today, kind of putative annotations, at least from a particular class of annotations that we're doing with tandem mass spectral libraries, and we'll get into the details of that a little later, we can get up to 13%. So it's not 100%. We still are quite a bit of ways from that, and there's some technological leaps on the horizon that'll we hope that'll get us, significantly closer to 100%. But this is the kind of state of play over the last 10 years, and we've made significant strides. And we're going to carry you throughout this presentation on some of the technologies, some of the approaches that we did that enabled us to have these big leaps. And so just to frame this in a pictorial form, imagine this gray oval is all the analytes that we can detect by mass spectrometry. We just use the public data as a proxy for what people are actually detecting out there. So there's about 1 billion tandem mass spectra in public repositories. And here, just first, we want to be able to re-identify known compounds. And this, again, can range between single digit to the teens in terms of the number of or the percentage of metabolites that we can annotate, and that's highlighted here in yellow. One of the approaches that we'll talk about is how do we start expanding this knowledge to this local neighborhood so we can start growing the space that we do know, as well as thinking about these other islands of unknown space that we can start prioritizing and pulling out for discovery. And so we'll show some examples of that. And so generally, the talk outline here is first, as mirrored in the previous slide, how do we identify knowns? How do we propagate and expand this knowledge from the known to slightly unknown? And then third is how do we start tapping into this unknown chemical space that may not have any known near chemistry that might be exciting for discovery. And so we'll start off with identifying knowns. And so just to highlight how this is done, one of the ways this is done, one of the more popular ways this is done, is you may have an unknown tandem mass spectrum here on the top left-hand side. And so one of the ways that we do this is we ask the question, has anybody ever seen this tandem mass spec before with a known structure? And so we ask that question in relation to a library of annotated MSMS spectra that have the spectrum and a known structure with it. And so this is called a library, and we ask this question across all the spectra within a library. And if there is something that is similar and it matches our unknown tandem mass spectrum, we can say, okay, we think they're very highly likely the same chemical entity. We can go to the library spectrum and say, what was the structure? And we remember that annotation, and we transfer that annotation and say, well, because our unknown tandem mass spectrum looked like this library annotation spectrum, we think it's this structure. So there's this cycle of inference based upon similarity in the tandem mass spec. Now, we've been doing this for the last 20 years, 25 years. Well, we go back to the literature, it goes back to the 60s, but kind of high throughput, we've been doing this for the last 20, 25 years. But one of the problems when we started really scaling this up is, where do we actually have the libraries? And how do we actually start building up these libraries? And so back in 2013, 2014, we borrowed some ideas from one of these some of the most popular crowdsourcing knowledge resources out there. One is the Protein Data Bank for 2D protein structures, or 3D protein structures, and Wikipedia for kind of does not need an introduction. And so we want to borrow these ideas from these communities and see if we can apply them also to the tandem mass spec library creation space. And we think we were relatively successful in this particular space. And so what we did was we built out these systems such that anybody from the community originally targeted to the natural product community is that they could come in with their tandem mass spectra and their structures that they laboriously solve their structures, or they had run a lot of standards and deposit in a very easy way into this community resource. And so this is the original idea and one of the original pillars of the GMPS platform. And so we started with just a few thousand back in 2014, and these days it's grown to a couple hundred, like 600,000 community contributed tandem mass spectra. So it's been, it's been very exciting that people actually want to contribute. And so one of the very natural questions whenever we want to think about how good or how useful is this particular resource? And so we want to ask about the reliability and the trustworthiness of the contributions from the community. And so one of the ways that we did this, when our original publication is we took this library and we searched a bunch of public metabolomics data. And so we ended up with putative annotations within this public data. And we went back to the community and asked, hey, this is your data. Tell us, does this make sense in the context of your understanding of this data set and your original publication with regards to the annotations and things like that? And so they could say, for each annotation, was it incorrect? Was it correct? And several levels in between. And so what we found when we asked this particular question is that on the two left-hand columns here were two specific GMPS libraries that we used for the annotation. And these annotations were as good or better than kind of state-of-the-art, highly curated resources that were used as a basis of comparison for annotation correctness. And so one of them was the National Institute of Standards and Technology, which is out of the U.S. Department of Commerce. And so that would give us some confidence that crowdsourced annotations or crowdsourced libraries were useful or could be useful for annotations going forward. It wasn't providing a lot of bogus annotations. And so that was kind of a good moment for us. But as we're growing these libraries through crowdsourced annotations, what we found throughout the years was, yes, we were growing, but we were not growing as quickly as we had hoped, right? So being much less than 1% in 2014 up to 2022, we were able to get to 22%. So that's great-ish. But I think my camera is not very happy with me. Just give me a quick. We will switch to. Okay. Well, we'll do it without the camera. We're still okay, Diana? On the audio, at least?
Yeah.
Okay.
The audio is perfect. Yeah. Thank
Okay,
you.
no worries. No worries. We'll do it without video. But we got to about 4% identification rates with the crowdsourcing, which is huge benefit to the community, but still we're not. There's how do we get to these step functions to have a path, a realistic path to 100%. So one of the things that we took advantage of is how do we think about propagating from this known space to this slightly unknown space and start expanding this knowledge so that every contribution from the community has an amplified impact, not just identifying the same compound, but starting to illuminate a local neighborhood. And so that's what we're going to talk about in this section, this step function at the very end. And so I want to give us a little bit of intuition as to how we actually got there. And so here at the very top, we have two structures that are very, very similar, right? They only differ in this lipid tail here. They extend it by a few carbons. And so this happens to be an antibiotic. And whenever we look at very similar structures and we put it into the mass spectrometer, we can ask the question, how do you expect the differences in the tandem mass spectra to be once you put into the mass spectrometer if they are similar in structure? And so very broadly here, we would expect in this particular case that they are very similar in fragmentation. And you can see just kind of, we won't look too details in this particular spectrum, but what we can see is the relative peak shapes and the intensities, they're very, very similar. And we just see some shifts and differences in the actual masses that result. So that's great. Now we're going to look at details on how that difference can manifest. And so in this particular toy example, just to simplify matters just a little bit, we have a molecule on the top and we have a molecule on the bottom. And they're mostly similar, except there's a change in structure that's exemplified by this little delta here. Now, my apologies to the chemists about this, but in terms of how crudely it's drawn, But the intuition that I want to get across here is if we imagine a fragmentation, the equivalent fragmentation from the top molecule and the bottom molecule highlighted with the blue dashed line, what we can see is on the bottom left of each molecule, they have this orange fragment. Now, they're the same because they don't have the delta. And when it's imprinted onto the tandem mass spectra, it results in this orange peak. And the key idea here with the orange peak is that it's the exact same mass. Now, if we look on the top right-hand portion, on the top molecule, it generates this purple fragment. And on the bottom molecule, it generates this blue fragment. Now, they're different colors because they're different masses. And the reason the bottom blue circle is different mass is because it adds this delta structure. Now, when it is manifested in the tandem mass spectra, the top purple portion is at a particular mass, but the bottom blue fragment is shifted. And the shift is exactly the mass of the delta. So there's kind of a consistency here. Now, this is one way that we can think about if you have the exact same or very, very similar fragmentation points, this is the generative process that can result in shifted peaks. Now, again, we know how much the peaks will shift if there's a single structural modification because we can weigh the two molecules and we can figure out the difference in the precursor masses, which is exactly the difference in the structure. Now, when we have these two tandem mass spectra, we want to be able to score them to account for this potential shift in mass. Instead of just thinking, traditionally with library search, we look at the exact same masses. We want to start thinking about how to actually account for this. And so the way we start thinking about this is for every peak in the top spectrum, it can appear at the same mass or at a shift of the delta. And so this is possible for every single peak in the top spectrum. And so we explore this entire space where should the first peak be at the same mass or shifted? Should the second peak be at the same mass or shifted? And we computationally explore this alignment space between the two spectra. And we find that answer for every peak such that it maximizes the similarity score between the top and bottom spectrum. And so this is how we can start accounting for these shifts and score pairs of tandem mass spectra. So this allows us to get to that point of every pair of tandem mass spectra, we can score it that is tolerant to these single structural modifications. But there's a little thing that pops up, right? So it's not perfect. So we consider here in this formulation single structural modifications. But the problem is in this particular formulation, double modifications cannot be aligned. And so we show an example here on the right-hand side where you have a delta on the top right portion of the molecule, and we have a gamma on the bottom left-hand portion of the molecule. And when we accounted for the shifts, we looked at the difference between the precursors, but not of the, if you have two modifications, they're added together, and so it doesn't align it the alignment is not possible. And so the way that we think about this is that biology and these actual complex mixtures offer us a potential solution going forward. And so the hope is that these modifications don't happen atomically. So there's some sort of biochemical process where you have the unmodified molecule on the left-hand side, it gets singly modified in the middle, and then it gets doubly modified on the right-hand side. So the alignment formulation allows us to do the left molecule, align the left molecule and the center molecule, as well as the center molecule and the right molecule, and then everything gets bridged together, and we can transitively align everything together nicely. And we don't have to leap from no modifications to two modifications. And so this is the underlying principle that we create this molecular networking concept. And so we can scale this up to all molecules within an experiment. This allows us to get around this double modification or multiple modification limitation. And just to give you an idea and a picture of how this looks in real life, this is a paper we published a few years ago, and this looked at the microbiome, the molecules produced by the microbiome of mice in a germ-free and when it's colonized, and looked at the differences in the metabolism there. And so we can see on the left-hand side, each of the circles is a unique tandem mass spectrum, which we think hopefully corresponds to a unique molecule. And these molecules are close together if we think that they're similar in structure because they have similar fragmentation. And so this is one small part of a molecular network. And so we can see there's quite a bit of diversity here. And what was interesting here was we focused on this particular region because there was one identified molecule here, which was glycocholic acid. This is a well-known bile acid that is being produced by mammals. And we were able to identify this particular metabolite, but there was all these metabolites around it that we didn't know what they were. And so this was an opportunity for us to annotate related compounds guided by the organization of the molecular network with a seed from a known compound in the libraries. And so this is the illustration and work done by Emily Gentry, who is now a faculty at Virginia Tech. And she's been a great collaborator. And so as we can see here, what she was able to do in this particular illustration is we looked at this neighbor that's directly next to this glycocholic acid that was differing by 90 Daltons. And so based upon the fragmentation differences, the peaks that shifted, she was able to figure out, okay, it's not glycocholic acid, but it's very similar. They replaced glycine with phenylalanine on the tail of this particular bile acid. And similarly, she was able to propagate this further. There's another 16 Dalton difference, and we were able to see another variant, which was the tail being replaced by tyrosine. And so these were interesting from a biology perspective, but from the chemistry perspective, the molecular networks were able to easily guide people and prioritize where the unknowns were relative to the known metabolites. And the organization prioritized these out of the tens of thousands of metabolites that were in the sample. And so that was pretty exciting from a discovery perspective. But one thing that, as a computer scientist that I don't like is that there is some manual effort on behalf of the chemists. And so they're manually annotating these peaks and trying to propagate these annotations in the molecular networks. Even though it saves a lot of time, we feel like we could approach this problem, define a computational problem, and solve it better to aid and accelerate discovery. And so this is what we did with Reza this past couple years. And so we wanted to make this particular process faster, so we formulated a computational problem around this. And so what we have here as a toy example, in the top is we have a known spectrum with a known structure. And so that, in this previous example, is like the glycocholic acid. And then we have a putative analog neighbor that is brought together by the molecular networks, and there may be a structural modification of some sort. And so one of the challenges is in the molecular network, we put molecules together and we say they're similar, and we give you a number, and that's about it. We don't tell you how they're similar. And so instead, so this formulates the problem. There can be a structural modification that is rotated at some atomic that is attached somewhere on the structure. So you can see these question mark puzzle pieces. They can be at any of the sites, but we don't know which particular site it is. So there's ambiguity that the molecular networks provide us. And so our job here is we want to figure out which one of these question marks is the actual correct site of the modification. And so the ModiFinder approach that we'll show just a little bit, the intuition is this. Whenever we actually do the alignments, a careful alignment between the known molecule, the known tandem mass spectrum, and the unknown tandem mass spectrum, there are peaks that shift and peaks that don't. For the peaks that shift, that gives us a clue that that is where that particular fragment, we think that's where the modification actually occurs. Because it weighed more, it should have one of the atoms where the modification attached to. Also, on the other side, if the peaks do not shift, that gives us a little signal it may not have that particular modification. So it gives us this inclusion and exclusion information about where the molecules actually are. or where the modification is actually localized to. And so the goal of the ModiFinder approach is we have this ambiguity of where the particular modification site is. And through careful analysis of the alignment between the two spectra and the differences between the two spectra, we can try to narrow down to one particular site or one smaller set of sites than everything. And so to give you just a small sampling here on a more real world molecule, if you have a particular peak that is shifted on the left-hand side that represents this purple circle, we say, okay, we think the modification is somewhere in this purple region. But if you have another peak that's representing the substructure in green on the right-hand side, and that's also shifted, the computation of the ModiFinder computational approach will take both of that information, and ultimately we try to score intersections higher, and that will lead us to think that the true modification site is actually at this intersection on this nitrogen here in this particular molecule. So we try to take all that information, synthesize it together, and create a particular prediction. And so I just want to say we'll have the workshop after this, and Reza will show you this, but the paper's out, and you can try it here at modifinder.gmps2.org. It's live. Go ahead and play with a few examples, and Reza will show you how you can apply this to your own data at scale as well. So that's the ModiFinder approach for this propagation. And so one thing we also wanted to do is once we can figure out this molecular networking idea and annotation propagation, what we also want, because we like to play with big data, right? The computer scientists, you always want to go big or go home, right? So one of the things we did was think about, can we approach this propagation idea not just at a single data set and help chemists analyze their own data, but can we start thinking about propagation at a repository scale? And so this is work with Wang, and he's now also faculty at the University of Antwerp. And what he was able to do was take 2,000 public data sets that's all published and in repositories, and what if we can create this mega molecular network, annotate the sites or the molecules that we do know, and then propagate one step out from that to look at these near neighbors to start enhancing the applicability of the libraries that we do have. And so just to give us an idea of what this resulted in, it was about 1.1 billion tandem mass spectra to create this giant molecular network. And then looking at confident nearest neighbors that occurred across many different data sets very consistently, we could explain the formula and all that, we ended up with about 88,000 putative propagated MSMS spectra that enhanced the library. And so once we added these to the spectral libraries, this resulted in about a 15% growth in the libraries, but about a 400% increase in the annotation rate. And that's what allowed us to get from that 4% to that 13% that we see in this particular chart. Again, this is not as high quality of a annotation as if you ran standards, but it helps create a hypothesis and guide discovery for the community. So that was pretty exciting that we could leverage the libraries and this big data to really start supercharging what we did know about all this public data. And so that's that kind of closes up that portion of the talk about how did we start expanding and supercharging our these libraries into the slightly unknown. But then what if we really want to start digging in to all this other public data that may not have any known metabolites to begin with? And so I want to introduce a new language that we created recently that is to mine public data, we think in a high throughput way. And so just to give us hark back to one of the figures earlier, we want to be able to start targeting these islands for discovery within this unknown space. So there's some applications that we've done on our side is maybe you want to look for aminoglycoside antibiotics. You can reasonably target something like that. You want to look for siderophores that may be implicated in the microbiome. You can do something like that with this new language. And so I want to introduce this kind of with a short story. So whenever Peter and I were really good friends, and great collaborators. And one of the strengths of our relationship is that Peter is a fantastic mass spectrometrist and chemist, and I don't have that sort of skill set. Rather, I'm a computational scientist, a computer scientist, and we have really, complementary expertise, and we just get along, love to work together. And so we were just kind of musing one of these a couple years ago. We have all this public data, and Peter asked, kind of a just off the cuff question. We have all this public data. Could we look for all carnitines in it? Pretty innocuous question. And so the idea is, I could think about that. I know a little bit about chemistry. I know that certain types of structures or compositions may yield distinctive fragmentation patterns, or just any sort of data pattern in the mass spectrometry data. So for example, if you have certain atoms in your molecule, it may yield unique isotopic signatures like iron or other halogens, or halogens in general. Or maybe you have a particular substructure in your in your 2D structure. It fragments in a very distinctive way that you could look for a particular fragment pattern in your data. And so I know a little bit about carnitines. I know there's certain neutral losses, certain masses that can occur. And I know a little bit about writing software. I can write a custom piece of software that will search for that specific pattern, scale this up, think about running this on clusters and start searching for this over hundreds of thousands of samples. So we can do that once, right? And we can give back the result. But the problem arises is that once Peter and other people in the community realize that this is possible, then they're going to ask for more. So there's a saying in English: if you give a mouse a cookie, he'll want a glass of milk. So I don't know if you're familiar with that. So but they'll just want to do more, as the basic line here. And they'll want to look for brominated compounds, look for siderosaccharides, peptidic natural products, drug metabolism, bile acids, and kind of the list goes on and on. There's a lot of imagination that the chemists have and what they want to look for. And being the lazy person that I am, I'm happy to do this once. I am not happy to do this twice, more than that. And so there's a part of me that's just wants to ask the question, can I develop a universal computational solution that empowers chemists to do it themselves, right? So, but they don't need to have the deep computational expertise to write complex software that can interface with mass spec data at scale and all this stuff, right? And so one of the things that we created that we think achieves this is the mass spec query language, or MassQL. And so the high-level goals for MassQL is that it needs to be understandable. So it's meant for chemists, so they should be able to read and write this language for chemists. It needs to be flexible, right? So it needs to describe everything that they actually want to find, from mass spec patterns to mass spec adjacent parameters such as retention time and mobility. It needs to be scalable. It's not helpful to anybody if you can just search for one spectrum. You need to be able to think about, hundreds of thousands, millions, and billions of spectra to search through. And it needs to be reusable. If you're going to take the time to write something in a new language, once you write it down, it codifies that particular knowledge and needs to be shareable across the community. And so just to give us a taste for all this, about flexibility and understandability, here is a small subset of what MassQL can do. Again, not particularly surprising, but again, this is a small subset for those of you who are not familiar with MassQL. So on the top left-hand side here, we can look for peak masses, right? Fragment specific masses that you care about in the MS1, MS2 spectra. You can find that here. You can look for losses from precursor on the right-hand side. You can look for gaps between two peaks within the spectrum, so neutral losses that aren't from the precursor, and you can look at relative peak intensity. Now, some of the power of MassQL is you can start combining any of these primitives and with conjunctions and have some sort of intersection between, and combine all this into a much bigger query. And you can also do things like mass defect, retention time, mobility across MS1, MS2, and things like that. So there's a lot of complexity that we're hiding here, but it just gives you a taste of some of the power that you could use. Now, if you don't want to, some of the newer technology that we've been leveraging with MassQL is for increased accessibility especially to people that are unfamiliar with MassQL. And so these days we taught some LLMs how to write MassQL. So if you don't want to learn the language, no problem. There's a little chat bot, write it in whatever language you want. It doesn't have to actually be English. And then it'll translate it into valid MassQL. And so it's actually kind of, I use this all the time. It's actually quite fun. So that's something you can definitely try out today. But just to show you one particular example with Daniel Petrus, he's a collaborator at UC Riverside. And so he cares about one class of natural products called albicidins. And so this is an NRPS PKase hybrid. And he published a paper describing a family or several variants of these albicidins in 2017 as part of his PhD work. And so he was able to very quickly write his own MassQL query because he knew that this family of compounds yielded distinctive fragmentation of 468 and 660 M/Z. And so that's the query we see here on the left, on the bottom left here. And he was able to apply this to the data that he had published back in 2016 in a matter of minutes and recapitulate all of his findings from that paper. These are the structures that he had published on. He was able to find them very, very quickly from his mass spectrometry data with MassQL. But more striking is that when he applied MassQL that he had written himself on his data and organized the subset into a molecular network, he was able to, in blue here, identify all the known albicidins that he had published on. But in gray, he was able to pull out a dozen new albicidin analogs that he had missed in his own data that he had manually looked through. And it was the specificity and the high throughput nature and the comprehensiveness that MassQL enabled for him to find exactly what he wanted to find that revealed new analytes that were relevant to him that he had missed because he was analyzing the data manually, and he couldn't scale his own effort that MassQL could take advantage of. And so this is one example where you can be more comprehensive in your discovery if you use these automated tools we hope that are easy to start leveraging on your own data. And so another example here is about scalability. And so with Daniel's example, it was not that big, right? We're talking about a few dozen files. It's not something you really want to look through manually, but when we talk about scale, we're really talking about huge repositories of hundreds of millions of tandem mass spectra, where looking through a few dozen samples is painful but reasonably doable manually. But when you're talking about thousands and thousands of samples, it's just impossible to look through manually. And so this is a paper that we published with Ipsita and Peter at UCSD about how we scaled up looking for bile acids in repositories. And so just as one example here, bile acids are a class of compounds that are being produced by mammals, or at least my understanding, mammals. And they can be modified in a number of ways by the body and by your gut microbiome. And so Ipsita, what she was able to do here is she knew a little bit about, or she actually knows a lot about bile acid fragmentation, and she was able to understand for this particular class of bile acids, subclass of bile acids, there is a 319 and a 337 M/Z distinctive fragmentation for this particular bile acid. And she was able to codify that as one particular query here. And then she wrote many others, but this is just one that we're going to highlight. And so she was able to write that very quickly and apply this easily to a huge amount of data. And so she searched over 100 million public tandem mass spectra. And this query was specific for trihydroxyl bile acids. And she was able to filter that down by 3 orders of magnitude into 270,000 tandem mass spectra. Now, with that in hand, she was able to take that and visualize it within these molecular networks and start understanding the diversity of the bile acids and exploring it in the context of a molecular network. And so this threw out, again, 99.9% of the data that wasn't relevant for this analysis, and was able to scale this molecular networking analysis specifically for bile acids across a huge amount of data. And this particular analysis, what it netted was there were 97 known bile acid modifications prior to this particular publication. But because she was able to process this data specifically for this class, she was able to confidently narrow this down to thousands of new observed tandem mass spectra that she thought was from bile acids with modifications. So this is a several orders of magnitude increase in the diversity of bile acids that weren't reported before. And what made this possible was the big data combined with MassQL, combined with her domain knowledge of bile acids. And so that's one area that the scale we thought was very, very helpful. Now, if you wanted to use MassQL, what we also wanted to do was make this whole platform and this idea universal, so you can learn one language and apply it across many different platforms. And so we have Python and R APIs that you can use. So this is open source and free for everybody, as well as integrated into a lot of these open source tools. And so GMPS is kind of the obvious one, but we also have it in tools like MSDial and OpenMS that these are open source free pieces of software, as well as commercial tools such as the Bruker's Metabolomic software. So you can use the same language in multiple locations for reproducible results and its effective data transfer. So that's one thing that's pretty exciting to us, that people were willing to integrate this into their own software. And so at the end of the day, what we've, asking that question, can we develop a universal computational solution that empowers chemists to do this themselves? The answer is, we think yes. At least chemists, people are doing this, writing their queries, applying it to data, and publishing papers on it. And the best part is, they don't bother me. So, I'm just joking a little bit, but it removes a key bottleneck of needing somebody on the team with extensive computational expertise, and we hope that removing that particular bottleneck can accelerate and increase the capabilities of chemists that are analyzing mass spec data. And so, just recapping where we were and what we covered here in this talk, we started out by creating libraries to annotate this known chemical space. We talked about the spectrum alignment and molecular networking and ModiFinder approaches that allow us to expand beyond this known space into near neighbors, shown here in these red arrows. And with MassQL, we were able to think about how we could start tease out and start targeting some unique chemical space that may not have any putative identifications or few putative annotations that come with a particular chemical hypothesis. And so those are kind of the three pillars that some of the stuff that we're working on in our lab, but we don't have time to cover all of them. But so that gives us some grounding there, as well as none of this work could have been possible without great collaborators throughout the years and throughout the world. Definitely some call outs are at UC San Diego during my PhD and postdoc work, and still close collaborators in Peter, Nuno, and Bill Gerwick, as well as fellow postdocs at the time, but now they're big shot faculty, people like Laura Sanchez, Vanessa Phelan, Trent, and Ben at JGI, as well as the mentees in my lab, the trainees who did all the hard work, as well as the funding that is very generously given to our lab. But happy to take questions, and we'll have 15-20 minutes hopefully with Reza to show you how you can use some of his new tool with ModiFinder interactively after this. And I think maybe we'll reserve questions afterwards, Diana.
Yes, we can reserve the questions after the Reza presentation. Thank you, Professor Mingxun, for your presentation. It was very interesting. I will pass the words to my colleague, Thomas Dussarrat. Thomas, please, the floor is yours.
Thank you. So now, a short introduction on Reza Shanheh. So the PhD student from Mingxun Wang, as well in the Department of Computer Science. So he joined the laboratory in 2022, and he's also using and creating computational tools to analyze mass spec data. So we already had a glimpse on what you did and what you are doing in the previous talk, but I guess we learn more about modifier that was introduced before. And yeah, Reza, the floor is yours.
Thank you for the kind introduction. Yes, so I'm just gonna go over how you can use ModiFinder, and just in my opinion, it is good to know how a tool works to be able to use it better. I'm not gonna go into like details, but I think a quick overview would be a good start. So for the ModiFinder, it has a few steps, the method. So the first step is that we are going to annotate the peaks of a known structure using in silico fragmentation tools. So the way it works is we start with a molecule. In the right, you can see a very simple example of a molecule illustrated as a graph. And then also we start with an empty set. And in each step, you are trying to add a simple fragmentation to this set. So each step, we are going to select an atom from this graph or a node from this graph, and then remove that. And then whatever remains from this graph is considered to be a fragmentation, and we add that to our set. So in this example, the atom A is chosen and removed from the structure, and then the remaining part is this structure. So we continue this step with the next atom. In this example, it's atom E, and the remaining is this, and we are gradually constructing our set of the fragments called S prime. And the next step is B. Once we remove this, we end up with these like smaller fragments. We add this. So once this algorithm is done, we have our fragments. And then we can just go ahead and measure the mass of these fragments and then figure out which fragment is assigned to which peak. Here is an example of this done on a real molecule and a real spectra. So you can see that after the fragmentation, the peaks are assigned with some fragments of the overall structure. However, we saw that this fragmentation is too general and ambiguous for our purpose. So what we did was we started using other known compounds in our libraries. So here you can see one example of this method being used. So on the top, it is our known molecule, and you can see the fragmentations at the top. But also we have this helper compound in our network. It is another compound that we know is similar to our known compound. We also know its structure. So we do the same fragment annotation for the peaks, and we see that like this orange fragmentation that we assign to this peak, if this peak is a shifted peak, that means that all its annotations are highly probable to contain the modification part. And in this example, the modification part is this red puzzle piece. So we expect the fragmentations to contain this modification. This is in line with what Professor Wang explained earlier, that we look at the shifted peaks in order to find these modifications. So in this specific example, we know both the compounds, and therefore we know where the modification happened. And in this case, it is at this yellow puzzle piece. Therefore, we can safely say that this orange piece was wrongly annotated for this peak. So because the annotation was in silico, it was not using that much of domain-related knowledge, and like it was not actually performing this under the conditions that a real molecule in nature would go through in its fragmentation. We might end up with like these fragments, like this orange puzzle piece, that are they're mathematically but not necessarily a good fragment to assign to this peak. And using these helper compounds, we can remove these fragments. So once this step is done, the second step is to find the alignment. And finding the alignment is exactly the way Professor Wang explained earlier. We just find the alignment that provides the highest cosine similarity score. And once this step is done, using these fragments that we assign to the peaks and the notion of the shifted peaks and unshifted peaks, we start creating this score for the atoms. So whatever fragment exists in a shifted peak, that would add a score to the atoms existing in that substructure. And if a substructure remains in an unshifted peak, that is a penalty, so a reduction in score. And this would give us a score for each atom that we translate to the likelihood of that atom being the modification site. So now we can just use the modifier, and I think with these examples, it would be even more clear how the tool works. So first, you can go to this website, modifier.gmps.org. And once you're in the website, you can click on this tab to open the examples and choose the second one, the neptomycin B and neptomycin A. I'm gonna wait just a few seconds just to make sure everyone's following this. And this is for you to have the website link. All right. So once you do that, once you click on this example, it would just automatically fill this part for you. But this is the part that you can fill with your own examples as well. So in this scenario, the USIs are the universal spectrum identifiers, and you can have this by just looking up these the molecules you have in the GMPS library, or if you have your own job run at the GMPS, you can just use the USIs provided for your job and your own data to pass your spectrums here. And also the second part is the SMILES, which is the text version of the structure of your compound. So in this example, because both parts are known, we do have the second SMILES, but in reality, that's the structure that you don't know, and it is okay. You can just leave that empty. This is an optional input. Once this is filled, you can just click on update, which would give you the prediction. In this specific example, as we had the structure of the unknown compound, it would also give you this like baseline thing to highlight the true modification. But in reality, if you don't have the second structure, you only get the prediction. So in this example, we see that like this part is the true modification, and the prediction is kind of relatively finding that modification, but it also is predicting different locations to be the modification site. Just one note, the more reddish an atom appears, that means it is more likely to be the modification site. So before I continue, let's just see what is this left panel saying. So the first information is the number of match peaks. As mentioned earlier, match peaks, whether they're shifted or unshifted, are a huge part in this prediction calculation, as we just rely on the information. Sorry, we rely on the information that the shifted peaks carries. So the number of shifted peaks is actually something that you can rely if it is a higher value, because that translates to more data. The second part is the number of shifted, and then the third part is the delta of the shifted part. I'm sorry, the delta of the weight between the unknown molecule and its known analog. So in this example, the delta is 14 Daltons. And it also gives you the modification type. In this specific example, it was an addition. So 14 Dalton was added to the known structure. So that's the structure of the unknown compound. And the last part, in this specific example, it will give you an evaluation score, and it is evaluating how good the prediction is. This is only available in the cases that you pass a structure for the unknown molecule. This is just an evaluation of the performance of modifier, and it's only possible if we have both structures to evaluate the prediction. So in a real world data, once you use this, you won't have this evaluation score because probably the unknown compound is unknown. And then this part is going to give you a structure, a predicted structure for that for this modification. So here it is predicting one of these two to be the likely modification that happens to the known structure. However, this can be improved if we can reduce the tolerance of the algorithm. So before it was set to 40 ppm, but if you change it to 20 ppm and click on update, we can see that now this possible modification is improved because the error tolerance is reduced, and one of the predictions that was only possible because the error tolerance was such a large window is now removed, and we are ended up with this smaller section of prediction formulas. So this ppm here, it is related to your data. You have to figure out how accurate your data is, but the more accurate you can set your tolerance error, the more accurate prediction you're gonna end up with. All right, so the prediction you see here is just modifier using its own annotation tools and alignment. However, chemists, they know more about their data, and also they have more knowledge than the current version of the software. So we have added this ability for chemists to add their own knowledge to the prediction. For example, if you scroll to this alignment part and click on peak 133, you see what it's been matched with, and also you see all the fragmentations, all the substructures we have aligned to this peak. So what this peak is annotated with. And then you can click on the edit part and remove the structures that are unlikely to be actually the annotation for this peak. So here we know that this amide bound is more likely to break. So this substructure is a more likely candidate for that peak in comparison to these structures. So once you deselect these and click on apply and do this for do the same for these two other peaks, 145 and 147, you can see that the prediction improves. So this is the prediction before, and this is the prediction after. If I was too fast on this part, I'm sorry. Once you click on the example part, there is a note section on the right, and it explains which peak you should select to modify things. If later you want to practice this yourself, just there's a note for you to go ahead and look at. Anyway, so once you do these steps and you refine these annotations based on your own knowledge, based on your own data about your structures, about your compounds, you end up with this hopefully better prediction in this scenario. These like other possible cases of modification is now removed, and the modifier prediction is highly concentrated in the correct modification part. And sorry, and I forgot to add this slide, but if you check the evaluation score, you see that it's been increased in this scenario. So this is how you can use modifier for your own data. If you want to use modifier for a larger batch of data, then you can go to GMPS. Hopefully you do have an account. Otherwise, you need an account. You have to create this account. So you can just follow these slides, and later on you can create your account and do it with your own data. But once you have your GMPS account and you've logged in, you can just go to this workflow section, search for high throughput modifier, and launch your workflow. So once you do that, you choose the name, and then you pass your own data. So if you're following this, set of instructions right now, if you do this, there is under the data set workshop modifier, there is a selected MGF file, but this is just an example. This is an example that is relatively fast to perform, so we can do it during this workshop. But otherwise, you can pass your own MGF file of your own data. So you can click on this select part, and that is your input data. Once you do that, you can also select the libraries. So by default, all the known libraries in GMPS are available for the library search part. But for this workshop, I've chosen just a very small fraction of that to make things run faster. So you choose this res.mgf, and then you add that to your library folder. But again, you can choose the whole GMPS known compounds library and use the benefit of this huge data set that is available through GMPS. So once you do these two steps, you should see the selected MGF, which is your data, in the input data, and then the rest MGF, which is the library folder in this second part. Again, for the first part, you can just use your own data, and then for the second part, you can just pass the whole libraries in GMPS. You then set this analog search to yes. This would help find compounds that are analog to your compounds in when doing the library search, and then you can just go ahead and submit the workflow. You probably see your workflow to be running. Once this status is changed to done, you can just go to this modifying the results. And this is what you see in that step. So it is going to give you some general information, but also it is going to give you a prediction confidence. So what you're going to do is you can sort the result based on prediction confidence. So you want to start with the data that is more likely to be correctly predicted by modifier. And this can have multiple reasons, one of which is the number of shifted peaks, as I mentioned earlier. So this prediction confidence, it is using multiple data to just assess how good modifier can perform. And so you want to annotate your data, and hopefully you want to start with something that is more likely to be correctly annotated. So you can search, start your search with just the ones that have higher confidence. Once you do this, you can just click on this view modifier. If you're following this workshop, you can just click on the one that has the scan number 24429. And once you do this, it just goes to the modifier website, which we covered earlier. It will go to the exact same service and gives you the prediction and you can just manually adjust the peaks and its annotation. Thank you.
Thank you, Reza, for your presentation. I think we have some questions. So the first one is, what is your future work? Are you going to use AI technology to this area? And how? Thanks.
Well, we can't give away too much information. Well, before, like, I think the answer is yes. But so stay tuned. Stay tuned, I think. It's just a lot of the stuff, as with a lot of research, before it's ready, you don't want to promise too much because nothing works until things work, right? So it's one of those things.
Okay, thank you, Professor. So the second question is, hello, thanks very much for an amazing talk. I did use GNPS, and after library research, many of the compounds are identified as Feinvolk-based bin. I looked for these compounds in the website of Feinvolk-based, but don't find the name of those compounds. Could you please provide any solution? Thanks.
Right, so that's a good question. So I think one of the just to give you an understanding, so some of the things that we did was we imported from the MONA, the Mass Bank of North America, into GMPS several stages of this, right? And so, you know, the earliest was probably like 2017 or so, some pretty old, a while ago. And so I think some of the problems that arose is the identifiers changed sometime in between. So that's causing these links to not work. So stop by our office hours. So we hold weekly office hours for the community. Don't email me about them, just show up. So there's an RSVP link. You can find it on Google for it, but this is basically what it's meant to. We can look at it because I get too much email. If you email me, you're not going to get an answer. So come to office hours is the perfect kind of place to kind of ask questions, and we can interactively look at something together. And on my side, it compresses. You know, I don't get bogged down switching too many tasks, and so that's the best time to look at this stuff in detail.
So another question, from 4% to 15% is a great jump. However, what is the main challenge to bump this number even higher?
Yeah, that's a good question. So I think one of the big things is so if we want to expand libraries, that's one way to do it, right? Is solving structures is slow, right? So, you know, especially totally novel structures we're talking about especially from the natural product side. So I mean, we have some collaborations in this area, is accelerating structure solving from the NMR perspective. So you know these totally new things, what it is. At the same time, there's only the other side of the coin is you can generate libraries of known standards. But creating these libraries, running them, it's expensive, right? It's synthesizing is expensive. And there's only a finite number of these standards out there that's well cataloged, especially for those that are relevant for these things. And so I think I'm not overstepping my bounds, but Peter has been synthesizing a lot of stuff. So he has an R01 for that. So I think he's just generating a lot more data in this particular space. So that's generating some ground truth data that's really, really important for an identification perspective, but also kind of underlying all this is how do we get good data to build new models that, like machine learning models, or just even understanding basics of fragmentation very comprehensively from a lot of good validation data. And so these efforts about scalable data generation are going to be in data structure solving. These are going to be huge pillars, the foundation for either understanding fragmentation really well going forward or building these machine learning models. And so there's a paper under review, and I think there's some really good work out in the field as well about defining these machine learning problems, having good, clean data sets. And that's going to be and onboarding new data as it comes in a nice way that makes this effort a lot easier and smooth. So I think those are the foundational things that will enable new computational approaches going forward. And so, we try to, I think there's so many smart people out there. We've been thinking about, it's like, we would rather work on building the good foundation for everybody else to make those cool computational leaps, like especially the machine learning side. It's just so many talented, and that's kind of our niche, rather than specifically competing. So it's we just want to be that complementing, being collaborative in that space. So we'll see. We have no idea, but that's how we were thinking about it, making a good foundation. And making the infrastructure, making the data solid. So long-winded answer. Long-winded answer.
Yes, thank you, Professor. So another question. That tutorial was cool, but really quick. Are more detailed tutorials available?
Yeah. So so Reza can answer them.
No, go ahead. I'm sorry.
And so my apologies. I was a little long-winded and took some time from Reza. So I think this was, we didn't intend for it to be like full, super hands-on. That we want to take like an hour or two just for the tutorial stuff. But again, I don't want to volunteer Reza's time, but if there's some particular applications that, in collaborations, reach out to Reza. I'm sure he'd be happy to see people using it in the real world. But also at the same time, just to get your feet wet, there are other tutorial videos on YouTube. We need to do a better job of collating all this stuff together in the documentation. But that's another place to kind of get your feet wet. But Reza, do you wanna comment on this?
Yeah, so one thing is that I understand that following this tutorial was a bit hard. It was too quick. One thing is that there are a bunch of notes across the website you can use, and it's going to guide you through these steps as well. Also, as Professor Wang explained, there are other videos available. In general, hopefully the website is easy to use. You can always email me if you're having any issues, and I'm hopefully going to answer you fast. And I'm looking forward to these emails, because as these feedbacks come in, I can just improve the tool with the specific use cases of people. This has been an ongoing improvement development so far with working with our other collaborators. So I'm very looking forward to any email coming from anyone using the tool.
Thank you, Reza. So one more question is, in fact, I was wondering about that. Can you identify microbiome-related metabolites in human blood samples?
So this is more of a biology question. My understanding is there are, but so one of the things we didn't talk about here is there's another tool that we introduced a few years ago, and there's new versions of it. It's called MASS. So it's a acronym, Mass Spectrometry Search Tool. Again, that's not very descriptive, but the idea is this. So we can take all the public metabolomics data out there. So from GMPS, metabolites, metabolomics workbench, all that together. And you can access it today. And what you can do is you can take a tandem mass spectrum and ask the question, which files does it actually occur in? And what we also paired with this particular thing, so you can do it in a few seconds to query across like 1 billion spectra and all that stuff. But the fun part is there's a lot of metadata associated with those files now. And once you have the metadata, you can see like which ones are blood, which ones are fecal, things like that. So you can understand what sort of relationship, which metabolites are present in one versus the other, or both, and things like that. So this is one way that based upon this sort of occurrence, you can overlay your intuition and your knowledge about, how do metabolites flow throughout your body, right? Do they go in the gut first, and you can gather evidence for that particular claim or hypothesis if you would like. So I think that might be a path forward to answer that particular question. But in terms of the actual biology, I'm probably the wrong person to answer that.
Yes, thank you, Professor. The one more question is, as a computer scientist working in metabolomics, how do you shift your skill sets from computer science to metabolomics?
Okay, so I'm also a bad person to answer this. So Reza, I will tell you. So Reza joined my lab when I started at UC Riverside. It's a little over 2 years ago, so it's very, very short. And so his background, well, he can describe it, but is pure computer science. And I don't think he'd ever heard the term mass spec before. But so he can tell you how his evolution. I'm personally impressed. It took me a long time to get on board with mass spec, but he can tell you his journey and how that went.
Okay, so first thing is that this is just data. Like once you get to it, it's just a bunch of numbers. Like, of course, it matters that what these numbers are representing, but like at the end of the day, it's just numbers, and computer scientists are good with numbers. So doing stuff with just that data is relatively very comparable to other aspects. It's just that once you start reading the papers, once you start collaborating with other chemists, and you gain some more insight about what these numbers represent, you can improve your work towards this very specific domain. But at the end of the day, it's just numbers. So it's good to be familiar with like what is everything, but don't look at it like it's a completely new field. You just need to know what these numbers represent, and it's like it's not that hard. The mentality, I think, should stay in a way that it's not like a new field and be scared to just dive in. But overall, my journey was just first like reading papers about what is happening in a mass spec, and what each of these peaks are representing, how we can like gain more insight, and doing things more in like a traditional way. After that step was done, I started like reading more papers that are computer science papers on like how this data is managed, handled, like shortened or converted to different like scenarios. For example, like a mass spec to a molecule. There is a like great field of research trying to convert a mass spectrometry to a molecule, and also the other way around, so a molecule to a mass spec. And I think reading these papers is also very helpful because they're trying to like adapt a bunch of machine learning scenarios to this very specific domain. And the way they use it, they're also computer scientist people mainly, and they also have to explain their thought process in their papers. So reading these papers like the iceberg, the mold discovery, like these papers, I think it is also a good approach. It's just prepare you for this transition.
And from my perspective, so to add on to this, is throughout my career, I think one of the best things that I've been very lucky to have is a good network, a good set of friends that are in the apply, they do metabolomics, they do biology, and they're just, we have a good time and we hang out, and they answer my questions. A lot of them are actually organic chemists. And so I think, especially like in my early days, like we sat in the same office with Vanessa Phelan and Laura Sanchez. I mean, they're faculty now, but back in the day, we were just kind of hanging out and talking about the science and just trying to meet in the middle and just speak the same language. So I think just taking that effort, I remember I went on Khan Academy and I learned some organic chemistry, right? So how do you identify like these functional groups, like just even having the nomenclature to do that, understanding these like SN2 reactions and all that stuff, right? So just trying to like learn that nomenclature and speak the same language, and having a good group of people that are willing to like teach you things that they're passionate about. It made it a lot more fun, and it paid off in the long term. So I'm very thankful for them taking that time to help me. And I mean, we had a good time too, but that really helped in my early days.
Thank you. So one more question. Can we somehow train AI to identify compounds? And also, how can your quality check the data deposit in the databases?
Right. So the first question is, and it's a loaded question. I think the answer is yes. But I think that's maybe beyond the scope of this talk. But at least the quality check of the data. So that's a pretty good question. So some of the stuff that we've been doing in order for the machine learning side, at the very least, is we want to make sure that the data going towards machine learning is clean and has the right metadata. And so number one, we make sure the metadata is coherent with everybody. So if we call an Orbitrap, right? But also, how do we do some basic checks about annotations? And so the very first one is, does the precursor match the structure? Does that happen, right? So questions like that, and starting to explain fragmentation and the peaks. Can you explain the peaks as subformula of the parent formula? Now, explaining it in terms of the fragmentation, the combinatorial fragmentation, that's, we don't go that far in some of these cases, but just making sure that that's not totally bogus whenever we enter the space of cleaning it up for machine learning. And so we published a paper in collaboration with Justin and Florian, in the Journal of Chemoinformatics, where it integrates a lot of this cleanup. So now if you go to there's the external.gmps2.org, you can download these cleaned up data sets. So this is run every 24 hours or so with the newest version of the libraries with all that stuff cleaned up. And then if it doesn't have structures, it's not included, and things like that. So these are some of the ways that we clean up make the metadata harmonized, as well as doing some of these most basic checks to make sure that the data and the structure, they are at least somewhat compatible, right? The obviously incorrect things. So those are some of the things that we do to make it reasonable.
We have another participant that is asking, I know it was not a part of this specific talk, but it is mole netting answer going to be implemented in GNPS2? I'm quite new to metabolomics data analysis, but I notice how this is used in many papers to merge results from different tools.
Yeah, we maybe not. I don't know. We can't commit to that. Maybe, but I think there's a lot of other things on our list. It's kind of interesting. Like, we want to support the community, right? But at the same time, we can't support everything. So we're still making decisions on what we can actually commit to. And also at the same time, right, you don't get grant funding for supporting older tools. But I will say, we'll do our best. No promises. But what I do ask of you all is, if you think like the resource that we're building, like GMPS2 and the legacy GMPS stuff is valuable, we're putting together an NSF proposal for the National Science Foundation here in the United States. Whenever we reach out to you, we might ask for letters of support from hundreds of people. You will say, yes, it is key to my work, and I'm funded by X, Y, and Z. And if it goes away, the grant falls apart. If you can say that, that would be fantastic. So that is no promises, but that's how we can keep these things going, I think.
Yes, I think it's a great idea. So please, to our audience, keep that in mind. And another question is, hello, thanks for the presentation. I want to know what is the difference between the using of compound discovered, cytoscape, and GNPS. Thanks.
Yeah, broadly, they're different tools, right? Compound Discover is from Thermo Fisher. So that'll be for only analyzing thermo data, right? So you can't use other vendors on that. So, I mean, they have a lot of cool tools in there, right? They have like feature finding, they have in silico annotation, they have some library search against MZ Cloud. So it's just a suite of tools that's published by the vendor. So I think if you like it, they have really good tutorials. It's not free, unfortunately. So Cytoscape is another generic tool for visualization of networks that can be used to visualize some of the outputs from GMPS. And so on the GMPS side, it's honestly like a million different things inside of there. So we have a lot of different tools for data manipulation, data organization. Some of the basics that we've highlighted here are molecular networking, but you can talk about library search, analog library search. You can do some sort of statistical analysis, other visualizations in it, ModiFinders in there. And there's targeted XIC, large scale targeted XIC. You can search all this public data. You can share data set, share like tandem mass spectra, draw for visualization. You can do box plot. So there's like a million different things you can do. You can even like make it really easy to enter author lists on Nature journals. Anyway, there's like a million different tools inside of the GMPS2 toolkit. So just check out our tool index and some of the things that you can do. But one thing I will say that we are not great at writing the documentation. I think we're not terrible, but I think we've only written documentation for like 30 to 40% of our tools. It's very common, actually. Some of our collaborators, like we built something a couple of years ago, and they're like, "Ming, it'd be great if you did this." We're like, "Oh yeah, we already did this. Yeah, here it is." They're like, "Why don't I know about this?" I'm like, well, so this is a problem. Maybe it's a good, it's a problem, but it's so I think maybe that's why office hours is such a good resource, so you can just ask. But anyway, so yeah.
Thank you. And so we have some more questions. So how do you deal with different modes, energies of fragmentation?
Right, Reza, do you want to think about this, or at least for the modifier side, you want to field this question?
Sure. Okay. So the current version of ModiFinder, it doesn't take into account the Gaussian energy, but it does not affect the performance because like different Gaussian energies, it breaks the molecule like in different distribution. However, like annotating the peaks with those with the substructures, it is not correlated to the Gaussian energy. It's just how the peaks are is correlated to that. So it doesn't need that part, but we have had like other research around this area, and we saw that like combining different Gaussian energies can improve the prediction performance. So if you have access to multiple Gaussian energies, you can improve ModiFinder's performance using that. But just the current version of the software, the Gaussian energy doesn't affect its performance because it can just annotate the peaks however the peaks are generated.
Okay, so I think I will give the word to Thomas. Thomas can do some more questions, please.
Yes,
well,
sure.
it's good. There's a lot of interactivity.
Yeah.
It's a good audience.
Talk all the time and you don't talk.
There's a lot of questions too. So the next question. Hello, thank you for your nice talk. I'm a PhD student in metabolomics, and I'm actually searching for a new strategy to detect the neural losses and the modifications of bile acids, but I didn't understand how I can use the MassQL for this object, and how it can help me to detect and annotate the modifications.
Right. So this is a pretty good question. So generally, MassQL, at least how we've used it, or I've thought about this, is you want to be able to capture the bile acids. So if you know something that's common to the set of bile acids you're targeting, that's how we've used it in the past, or at least I've thought about using it. I think definitely read the Cell paper. They go into significant details about specific patterns to differentiate different subtypes of bile acids. Now, if there's some novel modification that happens as part of once you capture the bile acid and there's some novel modifications, MassQL is not going to particularly help you there. So it won't be an annotation tool per se. It's more of a finding what you want to find tool at a large scale. Now, how to start figuring out the annotations of where it is or what the structure could be, that one of them could be used something like ModiFinder. So you capture everything with MassQL, and then you take a known bile acid and you put it in relation to a putative bile acid that's unknown, and you can ask the question, where is it, and things like that. So that's kind of how we were thinking about it. But check out the Cell paper. There's a lot of MassQL queries in there that do really subspecialize for bile acids. So then it's effectively some technology transfer. So you can learn from that and reuse exactly that to find what you might be looking for. So that might be a good way to think about it. And there might be some diagnostic peaks that Peter's group and Ipsita specifically did that is very relevant for particular hydroxylation patterns that will narrow down and be effectively an annotation aid for you. But they use it in a way for retrieval, but you can reuse that for annotation in your case.
Thanks for the response. The next one, are there a possibility to find new analogs from the chemical reactions through inside the mass spec and not but the sample?
I'm not sure I understand the question.
Me either.
Okay. Maybe we can move on.
Yeah, I think the next question. So thank you again from the great talk again. Now we are talking about DDA to get clean MS with the spectra. But what if we want to run DIA where we get chimeric MS2 spectra? In proteomics, there has been progresses to recompute putative DDA LAQV spectra from DIA data to use them to query libraries. Is anything like this coming in metabolomics?
Right, so that's a good question. So generally, you can do this with vendor software. Generally tackles this. So one of the ones that has the most complex DIA data is Waters. So their Progenesis software does do this, where you can get pseudo DDA spectra out. Now, there's another paper from Hannes Rost. He used DIA Met Analyzer or something like that. There was a Nature Methods paper, I think, about this, and they do also do a very similar thing that they do in proteomics, where you have the libraries and then you go back in, or the DDA, pure DDA spectra, and you can go back in and find where it is to identify and quantify in the DIA data. So that's something you can do. And also you can use tools like the proteomics tool DIA Umpire to generate pseudo DDA spectra output, and then you can feed that into a lot of our pipelines accept DDA like data, so you can run it in that perspective, from that perspective.
Thank you. The next question, is it possible to implement an independent library, for example, Lotus at GMPS?
Right. So you can deposit stuff at GMPS from any source, right? So we do this all the time for imports. So definitely reach out to us. There's some good documentation for batch imports, and it should be relatively straightforward. Now, I think what you're actually asking about is private libraries, especially GMPS 2. GMPS 1 doesn't support it, but GMPS 2, we don't advertise it, but maybe let's talk about this. Mostly we were trying to encourage people to actually deposit it and make it publicly available, so that it connects with this wider ecosystem, and you can visualize, you can search, you can do all this other stuff that requires it to be in the system. So that's kind of our incentive. You're just put it in this, but it's technically possible. So stop by office hours, and we can kind of discuss this. So we just don't want to support too much private stuff, because the whole ethos is making it open and all that stuff. But there are mechanisms that we take advantage of internally. So
yeah, I think that makes sense. But many people are still using a lot of in-house libraries, I think. The next question, is it possible to check the whole library of compounds using one small structure moiety like an amino acid to check its availability in different complex structure using the modifier?
I think this question is asking, given a moiety that they care about, tell me all the structures that it appears in. I think, and in the spectral libraries. I think that this is what it's asking. So if that is the case, you can do this without ModiFinder already. Actually, Reza implemented this. There's a library browser. It's called library.gmps2.org. And you can put in a substructure to search for, and it'll tell you all the MSMS that have it. And you can also ask the question of, what is the conserved fragmentation in the MSMS for spectra that contain this substructure? So that might be interesting to kind of think about fragmentation, consistency, and things like that. I think that's maybe what's going on in this question.
Thank you. Another one, are you working on metabolomics only or with connectivity on pathways and other omics, meaning multi-omics?
Yeah, so I mean, my background, my PhD was mostly in proteomics. And so we just, these days, or historically, we worked on these small molecule metabolomics problems, natural products and stuff like that. But yeah, so I think stay tuned. Hopefully some stuff will work soon. So you'll see some papers, but who knows?
Many things to stay tuned on.
Yeah.
So the next one I think is more for Reza. Is there a tutorial to create a USI? So I guess for MS Finder, modifier.
Oh, we do have a tool that creates USI from the like past data. I have to look for the link. Maybe Professor Wang has it like already in his like links. If not,
right,
you just send me an email. I know I have it somewhere. I just have to find it.
Right. So there's two things to think about, at least in my opinion. Almost everything, if your data is public or on GMPS 1 or GMPS 2, it's kind of like, it's like if you live somewhere, you don't ask for an address. You have an address. You just need to figure out what it is, right? So it's kind of that. So if your data is public or on any of these Metabolomics Workbench, metabolites, GMPS, GMPS2, massive, you can get a USI. And so you just have to figure out, how to get, figure out what it is. You're on the system. Now, if your data is not public anywhere and you don't want to share a whole data set, there's a tool called that we created. It's called TinyMass. It's a play on tinyURL. Anyway, so if you're familiar with that. But you can make a single tandem mass spectrum into a USI just by pasting precursor and the peaks, and then you can use it throughout the ecosystem. So those are two possible ways to do it. But what Reza's saying too is there's some workflows, I think, that will make it easy for private whole data, like a whole spectrum or a whole spectrum file. You can get USIs for every single peak and things like that. So.
Oh, okay. And so for just following on this question, because I also had it, if you have a single compound, you can use, what was the name of the tool you can use?
Oh, so TinyMass.
Yeah, just TinyMass. Yeah.
Yeah, so tinymass.gmps2.org, something like that. So then you can create a USI and then it'll make one single spectrum public. It's not great for high throughput, but if you just want to like do one small thing to search for it, or any of this stuff, it's pretty convenient.
Thank you. So in which library hosts xenobiotic metabolomes and environmental pollutants?
That's a good question. So if you go to the GMPS, there's the libraries page. There's like 40 different libraries. So definitely read through the text there to find what's relevant to you, and you can select any of those subsets to search against your data. So that's probably the best way to go. I don't actually know any of them off the top of my head anymore. It's just grown too large.
So the last question was just thanking for the talk and asking if it was possible to have the email of Professor Mingxun Wang.
Yeah, Google my name. I am the only I am the top page on Google. So that's it's all over there. So that's very easy to find. So
And I think it's all for the questions. At least I don't see more.
Yes, I think it's all, Thomas. So I would like to let the participants know that this webinar is being recorded, and the recording will be uploaded in the Metabolomic Society website, and it will be available if you want to listen in the future. And also, if the participants have any other questions, please email us and we will direct your questions to our speakers. I would like to thank to Professor Dr. Mingxun Wang and Reza for being here today, for accepting our invitation. And thank you for the outstanding talk and for your availability. And thank you, Thomas, for assisting me.
thanks everyone. Thanks you two for the kind invitation. So
thank you,
really
Professor, and for accepting on short notice. Thank you.
All right, take care, everyone.
Thanks
Have
for
a
my
good
part
day. Have
too.
a
It
Monday.
was a very nice talk.
Thank you.
Both of you.
Have a nice night.
Ciao, ciao.
Bye. See you.